B01 in our course B01 in data management. And now we're going to ask ourselves some questions. So you're asking which of the following are true? So the first question is, open science relies strongly on the internet. Is that true or false? Second question, open access eliminates publishing costs. Third question, open data facilitates reuse. Open data increases confidence in research findings. True or false? Open access permits the whole society to benefit from scientific findings. 6, citizen science engages the public in the research process. 7, release of public datasets is important for career progression. F in FAIR stands for free. And you can think about how, if you think those are true or false, and then we'll go through them in class. We have more here, 9 to 16. 9, only figures presenting results of statistical analysis need underlying numerical data. 10, sharing numerical data as a PDF in Zenodo is fair. 11, sharing numerical data as an Excel filing via GitHub is not fair. A group website is a good place to share your data. 13, data should always be converted to Excel or CSV files in order to be fair. A DOI of a dataset helps in grading credit. Fair data are peer reviewed. And 16, fair data accompany a publication. To continue this practical on fair data, you can bring your own data set. So bring your own data sets. That's the most ideal situation. Perfectly a structured data set is probably the easiest to start with. You can share the person next to you, but ideally, if you bring your own, that's the best way to explore. And your thoughts for the class are how to make this as fair as possible. And think about those steps of what you need to do to align with findability, accessibility, interoperability, and reusability. How would you do that? And that's something that we'll talk about in the class. So it's very important that you bring your own data set, and we'll go through that in detail. So we want to define the metadata associated with our data, create a data dictionary to link to that. We want to make it accessible. It could be in our database. It doesn't need to be password encoded or anything like that, but it should be a primary publicly accessible database. It should be interoperable, not using any zero language, and reusable. So it should be applicable to diverse areas. An example of this is that we can look at say a FASTA file of sequence genomes. We create a data dictionary from that, talking about our time of isolation and so on. We can submit them to GenBank or to Figshare if they're suits. Interoperable and download them from the above and check it can be aligned. Reusable, how can new data be added to it? So you can do all these things. So what we're going to do is to just, for example, here is to download 4 foot mount SAT1 sequences of a polyprotein, create a README file associated with that, upload to Figshare, create a DOI for that, check it works, because sometimes it's not always clear. Download it and check that it worked. So let's do that now. We're going to do that now and open up our Chrome browser. So we're going to type in our search query. We're using publicly available data, but this is just an example. So we search for organism of interest. We're looking for SAT1, and maybe look for a polyprotein. Of course, we're going to get a lot of matches here. So what you might want then is ones of a particular size. So we look at a particular range, say 600 to 611 bases. So it finds hits there, and we can see that metadata is useful in terms of defining what they are. So we can send this to file, and what we can do is download their GenBank file. So we're going to download that. We're just waiting for it to work. And then we're going to download the FASTA file as well and create a FASTA file, README file based on that. So we have our 4 sequences here. These are FASTA format sequences that we've downloaded. And what we can do then is create some metadata based on these. So a simple way of doing that would be just create a new file. We're going to paste these in here. We're just going to remove the sequences. So this is maybe a very simplistic example, but it's just to illustrate that creating a data dictionary is maybe something, or not a data dictionary, a metadata file linked to these sequences, although the file, the information is embedded inside there, the headers of the FASTA file already. So what we can do is just wait for the computer to wake up. So we can just save this file locally in our directory. Where are we going? So we just call this readme1. And then what we're dealing with here, then they're all footnoted to these ATMOSAT1, polyprotein partial CDSs. So you put that maybe at the start. And just say where we got it from John Blank, and today's date, the 29th of August. And then what we can put in then is the sample names. So you can see the extension numbers have the extension number and the version.1 of the extension number. And they're just making the sample names to the extension numbers. So you can see what ones are present there now. And we have GenBank files as well associated with that too. So we just record those as well. And that is a very simple way of just recording a README file containing, just outlining what we have present here. Certainly for longer data sets, you might think of writing a way of doing this with code, maybe extracting the headers and then parsing out the information there using spaces or something like that. But this is adequate for our purposes here. We could, for example, include the lengths of the sequences and other things. But we're not doing that here because they're all the same length. So we put them in there. All length 609 base pairs. So that's an example of how we create a metadata file linked to a data set, and then we can put this on Figshare. So for example, we can go to a website like Figshare. Obviously, this is not primary data sets. We don't need to create a brand new data set. But we can go to the Figshare website. And if you don't have an account, it can be handy to create one there. We can go there, and what we can do is upload our data set there. So that's an easy way of doing this. So what we can do is create up a project for this data set and upload it there, and I can create a DOI. So we can create a new project, create a new item. We can upload our files there. So we have the two files containing the data and then our README file. So that's README1. And then we're going to add in the other ones as well. So you got the sequences here. So we're loading them. We're probably going to have more information in the file names, but it's just an example. So we're looking at foot and mouth disease /1 body protein CDS files from GenBank. Should say it's four of them. We can add on categories as well. So for example, we can say the biological sciences, and this is linked to evolutionary biology. You might be looking at, for example, host parasite, phylogeny and comparative analysis, for example. And then we want keywords, we want to sequences. Maybe not relevant here. And then a quick description, which we could add in more details, but we're just going to be brief here. Change the item type to be a data set. And we have the licensing here. So this means that it's freely usable by others for non-commercial reasons, and then keywords, sequences. For this, publish an item, publishing it, and we should be able to get our DOI here. So I have a copied link here. So we want to get the public DOI. And let's just check where that is. So it took a minute, but our DOI is now available. So we can copy that. And we paste it here. And then you can see we have a public DOI. We just go to that directly to that website. So sometimes the formatting of the fixture DOIs is not ideal. So our cursor is obscured by zoom. So we see there, that's what we just uploaded now. And you can see that they're visible there on the DOI, and then we can even download them. So we download them here, and then we can check that those sequences were downloaded, or we can kind of see them there, that they're downloaded. So that kind of illustrates how we can upload our data sets onto a database and then look at them when you download them. If we see here, we have our downloads, those sequences there we just downloaded. So that kind of illustrates how we can make our data findable, accessible, to an extent droppable and reusable, in a simple sense as for a simple data set.